So let's now proceed to the MOA and the physiological effects of glucagon. The effects of glucagon are mediated through binding to and activation of the glucagon receptor. The glucagon receptor is a seven-transmembrane G-protein coupled receptor, so it is also known as GPCR. It is predominantly expressed in the liver. It is also expressed in the kidneys, the adrenal glands, the gastrointestinal tract, and the pancreas. The glucagon receptor again belongs to a family of receptor known as the G-protein coupled receptor or the GPCR. So this diagram here illustrates the glucagon receptor. It is a GPCR type of receptor. So it is basically located on the plasma membrane or the biological membrane. So the main structure or the main feature of the GPCR structure is the seven transmembrane spanning segments. So we have here the seven transmembrane segments of the GPCR structure that is numbered from TM1 to TM7. And we also have here, they are also connected by three intracellular. So we have here from ICL1 to ICL3, and we also have here the three extracellular numbered from EL1 to EL3 loops. Members of the GPCR superfamily share the same basic architecture. So we have here seven transmembrane segments, or this is also known as alpha helices. We also have the extracellular amino-terminal segment and the intracellular amphipathic helix and the carboxy terminal. So we have here the diagram of our glucagon receptor and the mechanism whereby it will be activated in order to produce its physiological effects. So again, as their name implies, the GPCRs interact with G protein. G proteins that is located in the plasma membrane. So the G here represents the guanine. When an external signaling molecule binds to our GPCR, so in this case it would be the glucagon hormone, it will cause a conformational change in the GPCR. So this change then triggers the interaction between the GPCR and a nearby G protein. So what are G proteins? So G proteins are specialized proteins with the ability to bind to the nucleotides GTP and GDP. So when we say GTP, that refers to our guanosine triphosphate, and GDP refers to our guanosine diphosphate. The G proteins that associate with our GPCR are said to be heterotrimeric, meaning they have three different subunits. We have the alpha subunit, the beta subunit, and the gamma subunit. And they are attached to the plasma membrane by lipid anchors. So the figure here represents the activation of the G-alpha subunit. So this is the orange one here represents the G-alpha subunit of our G-protein coupled receptor. So this diagram here is the unstimulated cell, and this one here, not to the left, refers to our stimulated G-alpha subunit. So in the unstimulated cells, the state of the G-alpha is defined by the interaction with GDP. So we have here our molecule, our GDP, that is attached to our G-alpha subunit. And also the G-protein coupled receptor. So upon receptor stimulation, so the receptor will only be stimulated upon binding with the agonist to our receptor, and in this case our agonist is the hormone glucagon. So upon receptor stimulation, the state of the receptor will change. So this the G-alpha will dissociate from the receptor, and the G-beta gamma and the GTP is exchanged for the bound GDP, which leads to G-alpha activation. So the G-alpha then goes on to activate other molecules in the cell. So this is just the preliminary processes that is important in order for glucagon to produce its physiological effects. Here a diagram showing the overview of the mechanism of action of the glucagon hormone. So this occurs on the hepatic cell. So the first step in the mechanism of action of glucagon is when the glucagon will bind to our glucagon receptor, which is a GPCR receptor located on the plasma membrane of our hepatic cell. So the second step here, so when the glucagon binds to our glucagon hormone, the alpha subunit exchanges GDP for GTP, and after that, the alpha subunit will activate adenylyl cyclase, and the adenylyl cyclase will then activate another protein known as the protein kinase A or PKA. After that, the protein kinase A will phosphorylate the downstream enzymes, and it will activate the different catabolic processes in the body, such as the breakdown of lipids, the breakdown of glycogen, and the process of gluconeogenesis. So these are collectively known as the catabolic processes. The glucagon will also inhibit the glycogen, protein, and lipid synthesis, and these are known as the anabolic processes. So the glucagon has inhibitory effect on the anabolic processes. So long-term response will increase the transcription of gluconeogenic enzymes. So this diagram here again represents the processes that involves the MOA of our glucagon. So let's now proceed to the different physiological effects of glucagon, and we'll start with the carbohydrate metabolism, particularly on glucose production. So it is said that glucagon is important to increase the hepatic glucose production. It promotes the hepatic conversion of glycogen to glucose. That metabolic process is known as glycogenolysis. It also stimulates the de novo glucose synthesis, and that is known as gluconeogenesis. It inhibits the glucose breakdown or glycolysis and the glycogen formation or the glycogenesis. So this diagram here represents the regulation of the glucose metabolism by glucagon in the liver. So in this diagram, we have here our glucose, acetyl coenzyme A, and our glycogen. We also have here the different metabolic pathways that involves the formation and the breakdown of our glucose and glycogen and acetyl coenzyme A. So the red arrows here represents the metabolic pathways that are inhibited by glucagon, while the green arrows here represents the metabolic pathways that are stimulated by glucagon. So the glucagon increases the hepatic glucose production by stimulating glycogenolysis. So we have here our glycogenolysis, now the formation of glucose from glycogen, and gluconeogenesis, now the formation of glucose from non-carbohydrate sources. So this should be gluconeogenesis. So these two here, the gluconeogenesis and the glycogenolysis, are stimulated by glucagon in order to increase the production of glucose. While glucagon will inhibit the glycolysis and glycogenesis. So these two here, these are anabolic processes. The glycogenesis and the glycolysis are anabolic processes that are being inhibited by glucagon. So glucagon is critical in the life-saving counter-regulatory response to severe hypoglycemia. So when we say hypoglycemia, that is a low blood sugar, and a low blood sugar in a severe form would be detrimental, no, to humans or animals, and it can cause death. So the role of glucagon, no, is that it can be a critical life-saving regulatory mechanism in order to prevent the severe hypoglycemia. It is also a key factor in providing adequate circulating glucose for brain function and for working muscle during exercise. How about the effects of glucagon on fat metabolism? So glucagon promotes the formation of non-carbohydrate energy sources in the form of lipids and ketone bodies. Glucagon, it stimulates the breakdown of fatty acids and inhibits lipogenesis in the liver. In times of energy demand, glucagon enhances breakdown of fatty acids to acetyl coenzyme A molecules in the liver. Hence, glucagon contributes to a stable energy homeostasis during conditions where energy supply is limited, such as during fasting, or in the states of increased energy demand, such as during cold exposure. So these are the different physiological effects of glucagon. So first, it increases the hepatic glucose production. It stimulates the breakdown of fatty acids and inhibits lipogenesis in the liver. So aside from these, it also promotes the breakdown of amino acids. It also reduces food intake, and it increases energy expenditure. Lastly, it may also regulate the heart rate and contractility. We have here another diagram showing the organ-specific function or effects of glucagon. So we have here in the brain, it increases satiety and decreases the hepatic glucose output. In the liver, again, it increases the hepatic glucose output. It increases the oxidation of lipids and it decreases the lipid synthesis. In the kidney, it increases water reabsorption and glomerular filtration. In the adipose tissue, again, it has a catabolic effect. It increases the lipolysis. In the GIT, it decreases motility. And in the islets of Langerhans, it increases the hormone secretion. So we have here a diagram showing the organ-specific actions of glucagon. So we'll start with the main stimulus or the main stimuli for the production of glucagon by the alpha cells of the pancreas. So of course, the main stimuli or the main stimulus for glucagon production is hypoglycemia. We also have the glucose-dependent insulinotropic polypeptide and the amino acids or a protein-rich diet. So in the presence of these factors, the alpha cells of the pancreas will be stimulated to produce glucagon. So again, in the brain, it will, in terms of the food intake, it will decrease the food intake. It will also decrease the appetite and it will increase satiety. In the pancreas, it will increase the secretion of the hormone insulin. For the liver, it will increase the glucose production. It will decrease the glucose breakdown, increase the lipid breakdown, decrease the lipid production, and increase the ketone body production and the amino acid breakdown. So the glucagon, therefore, will promote the catabolic processes such as the breakdown of lipids, the production of glucose, and the production of ketone bodies. It will inhibit the anabolic processes such as the glucose breakdown, the lipid production, and the those two factors, which are uh, anabolic in nature. For the brown adipose tissue, it will increase the resting energy expenditure, and for the heart, it will increase the heart rate and the contractility.